And I'm from Institut Pasteur Paris, and actually I have an introduction slide. So you might have seen me yesterday as a person we tweeted 2 hours ago. I'm actually a computer scientist. I work on algorithms and data structures for processing what comes out of DNA sequencing. So alignments, assembly, and primary analysis type stuff. I worked on software for genome assembly, and also did a few genome assemblies of pictures you can see here. And this altitude thing is to tell you that somehow I'm a little bit out of breath sometimes, so if I speak slowly or stuff like that, it's because of that only. So the talk is about data structures for storing sets of Kmer sets. I realized that the title can be a little bit intimidating, so I simplified it. And actually it's really about searching huge databases of DNA sequences. And as an introduction, many of you have probably seen this slide. It's the decreasing cost of DNA sequencing. You've seen these slides too many times. It burns your eyes, so I've added some effect to it. And instead, I would argue that there is a more interesting slide to show you today. It's this one, which shows the growth of a database called SRA. It's a database that contains everything that has been sequenced and deposited in public repositories so far. It contains a lot of bases, as you can see, a lot of data, and it's been growing across the years. And now, in 2019, it's around 30 petabases. And the particular thing about this database is that it stores data on hard drives, but you cannot search within it. You cannot search sequences within it. And in fact, what you could do, what you would maybe want to do, is to search for sequences inside the database of everything that has been sequenced so far. And for instance, searching for transcripts, SNPs, gene fusions, splice junctions, and entire genomes, maybe. And to give you a perspective of what 30 petabases are, your laptop has 0.001 petabases. That's a terabyte. My institute has around 8 petabases of storage. NCBI SRA, the database I was talking about, has 30. So that's more than you can probably have in your institute. And just to give some comparison, YouTube is up there. It's estimated to be around 100 to 1,000 petabytes. So that's a lot of bytes. And I'm guessing some of you have already seen what the SRA looks like, but since this is a diverse crowd, I'm giving you a screenshot of the web page of this database. So it contains a list of sequencing data, and you can see that the list goes very far, to 100,000 pages. And each entry corresponds to a sequencing experiment. So here I'm showing just 3 sequencing experiments for human genomes using Illumina. And earlier I was talking about searching this database. So you might have noticed there is a search function here, but it doesn't search inside sequences, it just search for metadata. For instance, you can search Illumina, as I did here, human, and things like that. And just to give like an overview of what an experiment is really, so it's I clicked on the first one, and you get a set of reads, which are short DNA sequences output by a sequencer. And okay, they're colored by bases, but that's it. So it's really a storage of a lot of sequences. And in fact, there has been an attempt to search inside the SRA. That's what's called SRA Nucleotide Blast. So it's a blast, so that's great. You can search inside sequences. But the only minor inconvenience is that you need to select a single experiment to search within. So if you want to do a search across the whole database, you will need to press search 5 million times. That's not very practical. But it's a first attempt. And here I'm arguing what could be gained by doing large-scale sequence searches. So for instance, if you were able to search the SRA, you could search for conditions that express a certain isoform. You could do a survey of gene fusions across the whole cancer data sets available. You could search for a certain bacterial strain that appears in all previously sequenced metagenomic samples. And it's also important for antimicrobial resistance, because then you could search for everything that has been sequenced so far for certain drug-resistant genes, and also examine the phylogeny of plasmids carrying certain important genes for AMR. So essentially, this boils down to Blast, but for all of the SRA. This doesn't exist, but we would like it to exist. In order to define the computational problem behind it, you are given many FASTQ files, which are the results of sequencing experiment, the first one, up to 100,000 of them, and a certain DNA sequence S. And the question is, can you enumerate all the experiment identifiers where S appears? For instance, given a short sequence here, it can belong to 3 experiments. Number 23, which is a human RNA-seq, another one, which is another human RNA-seq, and another one, which is a human gut metagenome. So that's the kind of queries that we'd be interested to perform on such a database. This is just a made-up query, by the way. So there are multiple techniques for doing this, and I would like to pause just for a second and let you think about how to do this. So if you have some ideas, please raise your hand and shout them. If you were given this problem, how would you solve it?
Do you need to find every match, or just
that's a good question.
some matches?
Well, for simplicity, let's say every match. But in fact, you could settle for some matches. So,
Would you cut up all sequences?
I'm sorry?
Would you cut up all sequences and try to find matches?
Yeah, you could start cutting off sequences, and in fact, it's a great idea I'll talk about later on the back.
K-mers or patterns?
Yes, Kmers and patterns. In fact, it's the most advanced solution I will talk about. I thought of something a little bit more elementary. To be honest, I thought you would say this. And it's not such a, I mean, it's probably not such a bad idea, but let's examine it. So another possibility would be to do that. And I think a small company in the West Coast has on this, and it's called Google, and it actually does search across many documents. And it builds a huge dictionary. So that's actually a very valuable solution, but it's quite intensive in terms of resources, and we'll see why later. Another solution, sorry, I'm blocking the view of some of you. Another solution is to build what's called an FM index. That's a very powerful technique from computer science to search in a large text. And finally, much of you already jumped to what I think is the most advanced solution, is to do Kmer Search. And I'll talk about that later. But first, let's see, why wouldn't grep work? Most of you guessed it's because it's too slow. How slow is it? Here's the back of the envelope estimation of how slow grep would be on SRA. It has a bunch of FASTQ files compressed using gzip. It's going to be the bottleneck because you can only read at 60 megs per second on gzip. Fun fact, and this is a plug for another work, we actually developed a parallel gnzip, which can decompress much faster, and that's actually recent work. And so if you take gzip and you do a grep across the whole SRA, then a single file would take you 16 minutes. Thousands of them would take one day with 10 threads. And the whole SRA, which consists of around 5 million experiments, would take around a year using a cluster of 100 cores. So that's clearly not practical. Another solution, it's the fact it's a solution that has been used by search engines, is to build a huge dictionary that contains as keys the sequence and as values directly the answer for the search, so the list of experiments in which a sequence appears. So that's a very tempting thing to do, and we know how to build large dictionaries using hash tables. Only problem is the size. So if you want to index n experiments, each of them contain R reads. Each of the reads is of length L nucleotides. This requires this amount of memory, and each of the values are large. So essentially, too much space needs to be stored. And companies like Google have solved this by throwing a lot of computational resources at the problem. And in our instance of the problem, we cannot really do that because, first of all, we are not Google. And we want to try maybe do some nice algorithms and data structures to solve this problem differently. So instead, somebody said on the back that we could actually index only the small sequences, only small subsequences that appear inside longer sequences in the database. Could we do that? So if you were, for instance, to try to index all of the genomes only from proteobacteria, so E. coli and other bacteria, there's around 20,000 genomes, and it's not such a big FASTA file. If you concatenate all the genomes, it's around 100 gigabytes. If you count Kmers, so all the sequence of length k, inside this big FASTA file, you obtain 31 billion sequences, and that's more a manageable size of data to index. And in fact, my bioinformatics staff has done this. And we know from previous results that it takes around 100 gigs of space to represent. There is actually some very nice computer science behind this. And I worked on this problem to study what is the minimal amount of information required to represent a set of sequences. And 35 gigs is not so big, actually, if you compare to the naive way to store all of the Kmers, which would be 31 billion times 31 nucleotides. But that's not so large. But if you consider that it's only the genomes from a certain family of bacteria, then imagine scaling this to all of the SRA. This really doesn't work practically. So computer scientists have been using FM indices to actually search databases of sequencing data experiments. And this paper from 2017 has built a Burroughs-Wheeler transform, which is similar to FM index, on around 3,000 human genomes, not assembled, but the raw reads. And this took half a terabyte of space and a very large amount of metadata to store where the reads come from. So that's really kind of expensive if you want to scale this up from 3,000 humans to 5 million experiments. The good news, though, is that the Kmer search within this database is quite fast. So as far as I know, this is the largest effort to construct a FM index on sequencing data. So we'll move on to the solution that has been adopted by most of the community, which is Kmer Search. Many of you have probably heard what a Kmer is. If you have not, it's a sequence of fixed length k. So here, if you have a sequence, you can decompose it into many formers, or many Kmers of length arbitrary k. And we will do something that is called a membership query. So ask whether a Kmer is inside an experiment, is inside a sequence that is inside an experiment. That's a basic operation that enables more complex queries. So if you want to ask whether a large sequence is inside a sequencing experiment, then you can reduce to Kmer search by decomposing the sequence into all of its Kmers. For instance, if you want to search for a SNP corresponds to roughly k Kmers, all of the ones which overlap the base of interest. And so the problem of doing Kmer membership queries within a single experiment has been studied quite a lot and has been solved using many techniques. One of them is called the Bloom filter. And I will just shortly give you a flavor of what it represents. It's just a bit array, so this information displayed here, that records the presence or absence of sequences inside an experiment. And the way this is constructed is all of the Kmers that are present in an experiment are enumerated, and then they are inserted at certain positions in an array. By that I mean that a 0 is converted to a 1 at a position given by the Kmer. And there is some magic to do a correspondence between a Kmer and a position in this array, and computer scientists know that it's called a hash function. And then using such information, you can do queries. So you can say, this Kmer, does it appear? To answer this, you need to just determine its position in the array and just answer if it's a 1 or a 0. So we done actually a recent review on data structures that do this kind of queries. And also I talked about it last year at CGSI, so there is a YouTube video on this topic. But today I actually want to talk to you about a more complex problem, which is to do these queries not just for one experiment, but millions of them. So SRA, remember, has 5 million. And I think it's an open question on how to do this very nicely. You cannot just use a single Bloom filter for 2 reasons. If you were to construct a single array for the whole of SRA, then it would take petabytes of space, so quite a lot. And also you would lose the information of where the Kmer comes from. And if you were to construct one Bloom filter but per experiment, so you build 5 million of them, then that would be actually manageable. But then you would need to query each one of them, so 5 million queries. And it would take around 1 memory access, which is around 100 milliseconds times 5 million, so long query time. Not very satisfactory. So recently, researchers have found a better technique to do this type of operations, is to construct what is called a sequence Bloom tree. So what it is, roughly, is each experiment, here I'm showing 4 of them, for each of them a Bloom filter is constructed, and then a tree is constructed on top of them. This is to make faster queries. So actually, to be more precise, so each of the experiments here is just a single array of 4 elements. You can imagine that it contains 4 sequences or not. So that experiment here is summarized as a bit array, and it contains, let's say, 3 Kmers. This one contains 2 Kmers, and so on. So that's really a simplification of what it is, because in practice, each of the experiments consists of millions of bits. And then we might want to do a query to determine the presence of those 2 Kmers at those 2 positions inside all of the experiments in the database. So you can already see that for the Kmer on the left, it's present in all of the experiments. And the one in the second position is present in none. So this one. And the way the data structure is constructed is information is recorded at the root if it's consistent with the rest of all the leaves. So for instance, the root records a 1 on the leftmost position because in all of the leaves there is a 1 in the leftmost position. And same for the 0. So then a query can be done as follows. It starts by querying the root. And then since it sees a 1, it can tell, well, I don't need to search anymore because I know by construction that everything below has a 1. So I can already say, let's stop. This Kmer is present in all of my experiments. So that leads to faster queries. And same for the one where it's not present everywhere. So this is a neat data structure that has been improved recently by myself and mostly by collaborators. And the improvement is highly non-trivial because we are storing twice more information per node, and yet it takes smaller space. So just to give an intuition, then for the same example as before, you still have 4 experiments. And now the roots record a different type of information. It records the determination information that says, I know what is the value of these bits in all of the leaves. And it records how. Like, I know that it's a 1. Here I know that it's a 0. Here I don't know, because some of the leaves have 1s at these positions, and some of the leaves have 0s. The construction is recursive, so all of these intermediate nodes also record information at their levels, but for the leaves corresponding to them. Yeah.
So you're saying that the first one, if it's a 1, that means that you know everything below that position, right?
Exactly.
And if it's a 0,
if it's a 0
you don't know.
You need to keep going.
Okay.
So here, if it's a 1, you don't need to query this Kmer anymore. If it's a 0, you keep going to both leaves, to both children of this node. And here, you know that it's a 1, so you can stop for this subtree. Here, you know that it's also a 1. So the Kmer is present in all of these experiments, but in none of these experiments. And actually, there is a dash symbol that says that the bit at this position is actually not informative. It doesn't need to be stored, because as you can see, for those positions here, those 2 first on the left, then we don't need to store anything here because we already know the information from the top. So using compression techniques, we can actually shrink the information quite a lot and don't need to store all of these Bloom filters at the bottom. So we achieve fast queries and also compression. And to give some idea of how effective this is, on the left, so if you consider 2,000 experiments, it takes around 15 terabytes of space to store on disk. And the indexing structures, it has 2 iterations. The latest takes only 14 gigs, so 1,000 times less space. And you can store it quite efficiently, and also it resides fully on disk, and search is quite fast. I'll briefly mention also another technique, which is called BigC, which made the cover of Nature Biotech recently this year. It's from a group in UK, and what they did was index all of the microbial sequences which are present in the SRA. And to explain a little bit what they did, I'm going to roll back from a few slides ago, when I said, remember, searching within a million experiments, we can't use a single Bloom filter, and 1 Bloom filter per experiment would take a very long time. And actually, they did just this, and they found a way to make it fast. So what BigC is really a vector of Bloom filters, or equivalently a matrix of bits, where all the rows, each row corresponds to a Kmer, and each column corresponds to an experiment. And it's a matrix because all the Bloom filters have the same size. And this sounds a little bit weird because if you know about Bloom filters, you would know that the size corresponds to the size of the experiment. But here it's possible because they index only microbial data. And each microbe has roughly the same genome size. So it's fine to do it with a single Bloom filter size. And they use some nice representation tricks to actually do fast search. So a single search takes only a fraction of a second. And all right, fun fact, this technique is also what is powering the search engine Bing. So really used for something else. There are many other methods. So I just talked about a couple of them, and also plenty of other interesting data structures to talk about, which I won't have time. We are preparing a review of all of them, and there's also a short overview in the paper pointed below. And that's my conclusion. So sets of Kmer sets. So I did not actually define sets of Kmer sets, but it's self-explanatory. So it's a Kmer set is an experiment, and you're just storing a set of experiments. It's a very powerful representation for representing sequencing data. I would argue it's a really booming area in the small niche of bioinformatics data structures since 2016. So many researchers are working on it. Problem is that no method is really user-friendly yet. So we don't have a nice website that allows us to do nice queries, but I know people are working on it. So I talked about SBTs and BigC, and they have specificities. So far, there is not a single method that kind of works for all of the data sets, but I'm sure there will be in the future. And just to give you another perspective on the sequencing data that will be indexed later, so today we are on the petabyte scale, but it's projected that in the next decades we will have even more to store and sequence and search within and have much interesting insight from. So I think all of it is going to be useful. And so that's it. Thank you for your attention. I'll take questions.